CRISPR, the new GPS of medicine. What does CRISPR imply? Clustered, regularly interspaced, short palindromic repeats. Palindromes is a sequence of double-stranded DNA or RNA. The arrangement of letters read the same left to right. Repeats are short segments of DNA, 20 to 40 letters in length. Are palindromic in structure. The DNA transcribes into RNA. They're identically repeating. They're interspaced by spacer DNA. Spacer DNA fills in space between the DNA repeats. Each one is unique. Spacer DNA matches up perfectly with viral or bacteriophage DNA. Cas genes or CRISPR-associated genes make Cas proteins. There are two types of enzymes, helicases that unwind DNA and nucleases that cut DNA. What is CRISPR? CRISPR is an adaptive bacterial defense system. It can quirk the immune system of bacteria to edit genes of other organisms. That can lead to genetic engineering. CRISPR forms the basis for genome editing. It can target specific stretches of genetic code and edit DNA at certain locations. Why CRISPR? CRISPR is precise, cheap, easy, and quick. It is able to edit live cells and work forever in microorganisms. CRISPR can revolutionize everything from medicine to agriculture. The outcomes of using CRISPR could be curing retroviruses such as HIV and AIDS, prevent and cure cancer, prevent genetic diseases like cystic fibrosis, make designer babies, cure for mental illnesses such as Alzheimer's, reverse and or prevent aging, and even cure blindness. Ethnicality. Some think genetic editing should be encouraged, and some will discourage the idea. Certain areas may be encouraged depending on the person's perspective on curing diseases, eliminating aging, making designer babies, and that the benefits outweigh the risks. Some people discourage genetic editing because it goes against nature. The current problem with CRISPR is risks we do not know enough yet to avoid unpredictable consequences. Some technological advances are still needed. Ethical decisions along with regulations need to be made. Unknown errors can still occur. There is a possibility of accidentally triggering unwanted changes. The future, we already live in this world where CRISPR is being used. In reality, it's full of opportunities and changes. We most definitely will face CRISPR in the future. All we can do now is act carefully and respectfully as we advance, working on accuracy and monitoring methods with caution, reasoning, oversight, and transparency.